Hello, so we're going over my notes for cell biology and life science in a nutshell. We're learning a bit about peroxisomes and some of the reactions that go on. Currently, we're learning about a reaction that goes on essentially in plants, plant called photorespiration. So the um, Here it's showing the Calvin cycle in the chloroplast in the glycolate, so which is produced from the chloroplast, um, using the Calvin cycle, um, from a ribulose to 1,5 bisphosphate phosphoglycerate. So the glycolate goes into the peroxisomes. So using oxygen, hydrogen peroxide is produced, glycosylate is produced, glyoxylate, and glycine. In the mitochondria, glycine is turned into serine, release of CO2 and NH3. Serine goes from mitochondria to peroxisomes, turned into glycerate, which is then taken to the chloroplast. To 3-phosphoglycerate enters the Calvin cycle, producing ribulose bisphosphate, then phosphoglycerate. So this is a cycle, basically. So role of peroxisomes in photorespiration. During photosynthesis, CO2 is converted to carbohydrates by the Calvin cycle, which initiates the addition of CO2 to the 5-carbon sugar uh, ribulose 1,5 bisphosphate. Let's see. However, the enzyme involved sometimes catalyzes addition of O2 instead, resulting in production of the 2-carbon compound phosphoglycolate. Phosphoglycolate is converted to glycolate, which is transferred to peroxisomes, where it's oxidized, converted to glycine. Excuse me. Then glycine is transferred to mitochondria, converted to serine. Serine returned to peroxisomes, converted to glycerate, which is transferred back to chloroplasts. So this is called photorespiration. Okay. As already noted, the assembly of peroxisomes is fundamentally similar to that of mitochondria and chloroplasts, rather than that of the endoplasmic reticulum, Golgi apparatus, or lysosomes. Proteins destined for peroxisomes are translated on free cytosolic ribosomes to stay inside the cell, then transported into peroxisomes as completed polypeptide chains. Phospholipids are also imported to peroxisomes via phospholipid transfer proteins from their major site of synthesis in the ER. The import of proteins and phospholipids results in peroxisome growth and new peroxisomes are then formed by division of old ones. So the way peroxisomes grow is protein that are synthesized on the free ribosomes come into the peroxisomes, and new peroxisomes also can rise from the division of old ones. Proteins destined for peroxisomes are come into pre-existing peroxisomes. So, and old peroxisomes divide into new peroxisomes. Proteins targeted to the interior peroxisomes by at least two pathways, uh, which are conserved from yeast to humans. So these pathways targeting proteins. Most proteins are targeted by the simple amino acid sequence serine, lysine, leucine at their carboxy terminus. Peroxisome targeting signal 1 or PTS1. Other proteins are targeted by a sequence of 9 amino acids called PTS2 at their amino terminus. And some proteins may be targeted by alternative signals that have not yet been defined well. PTS1 and 2 are recognized by distinct receptors and transferred to translocation complex that mediates their transport across the peroxisome membrane. However, the mechanism of protein import into peroxisomes is less well characterized than the mechanism of protein translocation across the membranes of other subcellular organelles. In contrast to the translocation of polypeptide chains across the membranes of the endoplasmic reticulum, mitochondria, or chloroplasts, targeting signals are usually not cleaved. During the import of proteins into peroxisomes, cytosolic HSP70 has been implicated in protein import to peroxisomes, but the possible role of molecular chaperones within peroxisomes is unclear. Moreover, it appears that proteins can be transferred into peroxisomes in at least partially folded conformations rather than as extended polypeptide chains. Some peroxisome membrane proteins are similarly synthesized on cytosolic ribosomes and are targeted to the peroxisome membrane by distinct internal signals. However, other experiments suggest that some peroxisomal membrane proteins may be synthesized on membrane-bound polysomes of the ER, then transported to peroxisomes, suggesting a role for the ER in peroxisome maintenance. The import of proteins into peroxisomes thus appears to have several novel features, making it an active area of investigation. Interestingly, some components of peroxisome import pathways have been identified not only as mutants of yeast, but also as mutations associated with several human diseases involving disorders of peroxisomes. In such diseases, only a single peroxisomal enzyme is deficient. However, in other diseases resulting from defect in function of peroxisome, multiple peroxisomal enzymes fail to be imported to peroxisomes, instead being localized in the cytosol. The latter group of diseases result from deficiencies in PTS1 and 2 pathways responsible for protein import into the peroxisome. The prototypical example is Zellweger syndrome, lethal within first 10 years of life, results from mutations in at least 10 different genes affecting peroxisomal protein import, one of which has been identified as the gene encoding for a receptor for peroxisomal targeting signal PTS1. They contain okay. So we're gonna take a break. We'll continue in the next part, summing up the role of peroxisomes. inside cells. Thank you.